Greetings and welcome to the Tonix Pharmaceuticals Conference. It is now my pleasure to introduce your host, Dr. Seth Lederman, CEO of Tonix Pharmaceuticals. Thank you, Dr. Lederman. You may begin.
Thank you. It's exciting to be presenting Tonix to you on this digital format. We have a big group. I'm going to answer questions at the end, so there is a Q&A system, and please use it. I'll try to leave 5 minutes at the end to answer questions. So Tonix is a public company on NASDAQ, so I have to take you through our standard disclaimer on this next slide. I'll be making forward-looking statements, and our SEC documents are more complete, and we soon will have our 10Q to update financial information about the company. Tonix is a biopharmaceutical company. We are working on important problems. We are clinical stage. We focus on small molecules and biologics, and there are two main areas, CNS, or central nervous system, and immunology. In the CNS space, we have programs for pain, neurology, psychiatry, and addiction. In the immunology space, we have vaccines, immunosuppressive drugs, an oncology program, treatment for autoimmunity. Our full pipeline is laid out on slide 4. Well, this is part 1 of 2 of our pipeline. This is the CNS Portfolio, and in yellow is highlighted our fibromyalgia program, and I'm going to be focusing on the fibromyalgia program today. It's phase 3, and we're going to have interim data in September and top line before the end of the year. And on slide 5 is our immunology portfolio, and I'm going to be focusing on the COVID-19 vaccine that is listed, that's highlighted in yellow at the top, TNX 1800. And that's a preclinical program, but because of the excitement and the urgency of developing COVID-19 vaccine, it's a prioritized program. And I'm actually going to begin with that program. So TNX 1800 is a vaccine candidate to protect against the virus SARS-CoV-2 that causes COVID. 1800 utilizes our proprietary Horsepox vector. It's designed to express a protein from CoV-2, and we are collaborating in the development of the vaccine with Southern Research. In the middle bullet, we have announced a manufacturing agreement with Fujifilm Diosynth, and they are engaged to make GMP, Good Manufacturing Practice, vaccine for our human trials. The key milestones in this upcoming are in Q4, which is next quarter, and that will be small animal and non-human primate studies, including a challenge with CoV-2. The CoV-2 vaccine, the landscape is rich. There are more than 150 vaccines in development, but we expect, and many others expect, more than one vaccine will be approved by FDA, and we think that different vaccines will be selected by different doctors for different individuals. We are one of only about 5 live attenuated vector systems in development, and we have 2 such programs. I'm going to focus on our Horsepox program, but we also have a bovine parainfluenza program. Some of the other prominent attenuated live vectors are Merck's 2 programs in measles and VSV, and then an Indian company, Zydus Cadila. CoV-2 is challenging, to say the least. I think this slide is interesting in terms of comparing the 2003 SARS with the 2019 CoV-2. And the biggest difference at the bottom is that the 2003 SARS killed 744 people across the globe, whereas the current COVID has already killed, I believe now the number is over 600,000 people. So clearly SARS-CoV-2 is more deadly globally. But if you look at the rate of infection, people infected with the SARS virus died at an astonishing high rate. About 10% of people who were infected died. The rate of death for CoV-2 is much lower. It's estimated to be something like 0.4%. But I think you can see from the toll that it's taken that while it's less deadly per person, it is much more infectious. So the transmissibility is much higher. And all of these features really demand a vaccine that matches the threat. So we're using Horsepox because it can be engineered to express foreign genes, and in this case we're expressing a protein from CoV-2. And it has a lot of appealing features. It doesn't persist or get integrated into the host genome. It has strong immunogenicity. It can be manufactured at scale. And because it's a live attenuated vaccine, it triggers T cells, which provides a lot of durable immunity, at least to its own antigens, and we expect it will to this passenger antigen. And the passenger antigen is described on slide 10, where we've inserted a gene expressing the full-length SARS-CoV-2 spike protein into Horsepox, and that is TNX 1800. 1800 is designed, this is shown on slide 11, it's designed to be given into the arm like a traditional smallpox vaccine, and once it gets into the arm, the TNX 1800 virus replicates, infects cells in the skin of under the arm, and the virus also causes cells to express the spike protein of CoV-2. And the reason we believe this will provide immunity is that it exposes the immune system to the spike protein of CoV-2, but it doesn't have the same deadly potential as a full CoV-2 virus. So we're using Horsepox to introduce a protein from CoV-2, but without all the risk of the CoV-2 virus. Now, our vaccine is designed to stimulate T-cell immunity. And this slide contrasts T-cell immunity with antibody immunity. T-cell immunity is durable. It's long-lived, many years, and T-cells clear infected cells. So they wait till a cell is infected and then they kill it. Antibodies, which has had much more focus on it in the media and in scientific presentations, antibody immunity is actually quite short-lived, 3 to 6 months. It recognizes pathogens directly, but it's not the traditional way that vaccines for viruses operate. So we expect that our vaccine will elicit both T-cell immunity and antibody immunity, but we think that the T-cell immunity will be the distinctive feature and quite a bit stronger. The status of this program is that in the Southern Research arm of it, there are two key questions. Will vaccination of animals elicit an immune response to the S protein? And the second is, will the immune response protect non-human primates against the challenge with SARS-CoV-2 virus? So we're expecting to have answers to those two questions in the fourth quarter of this year. And while we're waiting on that, manufacturing development for GMP virus, that's the kind of virus that we can use in a clinical trial, is underway. We recently announced a partnership with Kansas State University to develop a second CoV-2 vaccine. This one is based on bovine parainfluenza virus from technology that was developed at K-State. And this has been widely used. Bovine parainfluenza virus has been widely used as a vector in the past. And we are excited about this program because it is also a live replicating program. So we have two different live replicating programs. And we think that live replicating programs have the strongest possibility of inducing long-lasting T-cell immunity. The live attenuated viruses, as I said, elicit durable immunity. They can be delivered on a single dose, which I think is very important, and the manufacturing is scalable. And that's because some of the replication of the virus goes on in the arm after administration. And finally, live replicating vaccines have really been shown to block forward transmission. And we think that this is a key to eliciting herd immunity and protecting immunocompromised people who maybe can't get the vaccines themselves. So now I'm going to change gears and talk about our Phase 3 program. This is TNX-102SL. It's a proprietary formulation of cyclobenzaprine. It's administered under the tongue or sublingual, and it improves sleep quality. So it has the potential to treat a number of different conditions where sleep quality is an issue. There are a number of benefits of TNX-102SL over oral formulations of cyclobenzaprine. Oral cyclobenzaprine is a swallowed tablet, has been available in the United States for over 40 years. It's approved for short-term administration for muscle spasm, and we're using it in a very different way as a long-term bedtime treatment for fibromyalgia and potentially some other conditions. So our formulation, the 102SL formulation, is specifically designed to provide this kind of benefit as a long-term bedtime medicine. In fibromyalgia, we've completed 2 trials, a Phase 2 and a Phase 3 trial. Both of these trials missed statistical significance in the primary endpoint, but in both cases, we've looked carefully at the data, and we're excited about moving forward, and I'll explain why in the next few slides. The safety program that we've developed, the safety package that we've developed from these 2 trials and from other trials is quite extensive. And I think it shows that cyclobenzaprine is well tolerated and that the benefit to fibromyalgia patients should be worth the risk when it's FDA approved. This is a summary, slide 19, of the 2 trials we've done. In the light blue column is a Phase 2 trial, and the darker blue column is a Phase 3 trial. And you can see below in the green, those are all the key secondary endpoints, and the green is highlighting the fact that many secondary endpoints were less than 0.05 p-value. Unfortunately, in both these trials, we missed the primary endpoint, and those are shown in yellow at the top. So we think that this indicates that what we were working on is a little bit too low of a dose. And as you can see, the dose here is 2.8 milligrams. So the way we've adjusted it in our program is to increase the dose. But before I go to the increased dose, I just want to show you that even at the low dose, looking at the primary endpoint of our ongoing Phase 3 study, the treated patients in light blue had a statistical improvement at week 12 over the placebo patients in gray. So even at the lower dose, using the measurement that's the primary endpoint of our current Phase 3 trial, we had a p-value less than 0.05. This slide graphically shows where we think we are on the dose-response curve. That the arrow pointing to that with the box inconsistent response, I think, characterizes where we were with the 2.8 milligram dose. And the arrow pointing to the orange circle, the box saying consistent response, is where we think we are with the new program where we've doubled the dose. And it took us a while to develop the data to double the dose, and we didn't get that data from the fibromyalgia program, but we have a PTSD program, post-traumatic stress disorder program. And the next few slides will show you that in the PTSD program, doubling the dose increased the activity of the drug. So here are 2 examples. where gray is placebo, blue is the low dose, and yellow is the higher dose. And I think you can see that in the CAPS-5 and the CAPS-5 sleep items, these are PTSD endpoints, that doubling the dose increased the activity of the treatment. And similarly, this is two other endpoints, the Sheehan Disability Scale, the same coloring, so the work-school item and the social leisure item both improved with the doubling dose. And then finally, I'm showing you the CGI-I, which is Clinician Global Improvement, and PGIC, Patient Global Impression of Change. And in both cases, doubling the dose in the PTSD trials improved the activity. So we're hoping and expect that doubling the dose will get us into a consistent response range with our PTSD trial. The tolerability of the program in PTSD, we think, was very good. In orange is shown the systemic side effects, which were lower in the phase 3 than the phase 2, but even in the phase 2 were, I think, showed a good pattern of tolerability. And in terms of local side effects, there's some mouth numbness that happens episodically in about 40% of patients, but it's self-limiting and goes away. So here's the phase 3 study that we've announced is completely enrolled, and this is the study where we expect to report data relatively soon. The most important change from the last phase 3 study is that we doubled the dose, but there are a few other changes. For example, the primary endpoint that is more suited to how the drug performed at a lower dose. So to summarize the first two programs, and this is really where I'm going to transition into summarizing it, because I don't really have time to take you through our pipeline. But the 102SL for fibromyalgia program is in phase 3. The RELIEF study is fully enrolled. We are using twice the dose of cyclobenzaprine in this trial than in our phase 2 and our earlier phase 3. We expect interim results next month, so that's September 2020, so it's really just around the corner, and we expect top-line data before the end of the year. The other program I took you through is the TNX 1800, which is the vaccine for COVID. It's preclinical. It's based on a live virus vaccine on our Horsepox vaccine platform, and in this program, we have two very near milestones. The fourth quarter, we're going to announce the results of both small animal response and primate testing. And those data we expect will also include the results of a challenge with COVID-2 virus. So before I wrap up, just a word that, we believe that the 102SL platform can be extended to a number of different indications. This drug, we believe, works by improving sleep quality. And just this morning we announced that our IND was cleared by FDA to study TNX 102SL in alcoholism. Alcoholism is now called alcohol use disorder. But in the past, we have already gotten approval to study this for agitation in Alzheimer's disease, which is an important consequence of Alzheimer's disease. And frankly, one of the most common reasons why people with Alzheimer's are institutionalized and fail home care. So I'm going to jump ahead to slide 34 to just summarize our platform. So you can see these four areas, pain, psychiatry, addiction medicine, and neurology. And the pain program is obviously led by our phase 3 102SL program for fibromyalgia that has very near-term data. But we have a number of other programs, including under addiction medicine. TNX 1300 has FDA breakthrough therapy designation for treating cocaine toxicity, and then the other part of our pipeline. Sorry, let's see. We call public health and biodefense, is led by our COVID-19 vaccine, TNX 1800, that I described, but we also have programs to protect against smallpox and monkeypox and radio protection. So the last slide I'll present, this is a very exciting time for Tonix. At the top bullet, we said we submitted the IND application for 102SL for alcoholism, and as I told you, the press release this morning announced that IND was cleared. In September, we're going to have interim analysis from the 102SL fibromyalgia study. Fourth quarter, we're going to have top-line results from that study, a phase 3 study, and we're going to have 2 important data points for the COVID-19 vaccine. So with that, I'm going to turn to the Q&A slide, but I want to thank everyone for their participation. So let me just get oriented because I'm not that familiar with this platform. The vaccine. Why are some other companies far ahead of us in the vaccine? Some are already some are already testing on people. And so the answer to that is, because we're focused on T-cell immunity, it is just harder and takes a longer time to develop a vaccine that stimulates T-cells. For those of you who have been following, the media or even the scientific literature, there are numerous articles about antibodies. There are only about 4 articles yet on T-cell reactivity in COVID-2. So T-cells are harder to measure. There's not a standard clinical laboratory system for measuring them. And, what we're doing just takes a longer time. But, we are in a similar boat to, for example, Merck, which is working on 2 live replicating vaccines. And similarly, they're not in clinical trials yet. And they are also focusing on T-cells. And as I said, one of the advantages of T-cells is that they provide durable immunity, whereas vaccines that only stimulate antibodies, might need to be, there may need to be boosters frequently. Okay, the next one. Are we seeking government funding for our COVID vaccine? Yes, but, we are looking at there are a lot of different potential funders in the government. The one that's gotten the most attention is Operation Warp Speed. And for Operation Warp Speed, I think that's really focused on 8 candidates, 6 of which have been named, which are able to provide 100 million doses by the end of the year. So as we said, we are in GMP manufacturing of our vaccine, but we are not in a position to provide the government with 100 million doses by the end of the year. Just going through. Well, okay. Well, I'm afraid that we're running out of time. So I want to thank everyone for participating and turn it back to the organizers. I'm sorry I didn't get to all of the questions, but I'm really grateful for the very large audience and enthusiastic questions. So thank you very much. Thanks for following us. And this is Seth Letterman from Tonix.
This concludes today's webcast. You may disconnect at this time. Thank you for your participation.